Look at how beautiful the seafloor is. This is a purely blue world with colorful fishes, moving in and out of different kinds of rocks and aquatics. But did you know the deep oceans, Earth's largest ecosystem, are especially energy-deprived system? With frigid temperatures and virtually no in-situ productivity, the deep ocean floor is really limited in energy. Today, we are going to present energetics of life on the deep seafloor by linking energetics across multiple scales of biological organization in the ocean. Life requires energy. The flux and the transformation of energy influence process and the patterns across levels of biological organization. Genomic, phenotypic, and taxonomic diversity and complexity are correlated with variation in energy availability in space and time. Variation in metabolic rates and life history traits reflects variation in temperature. The increase in body size and the increase in proliferation rate are both driven by increase in energy. As we all know, in deep water, light availability most limits productivity among all of the abiotic factors. This is all because water absorbs and scatters light, so the amount and the types of wavelengths available to organisms change dramatically as the depth increases. In the deep oceans, which encompass depths below 200 meters, limited light availability would lead to two results: low temperature and non-existent photosynthesis. Globally, temperatures of most of the seafloor vary between -1 and 4 degrees Celsius. These cold temperatures limit biochemical kinetics of metabolism. Due to lack of photosynthesis, primary production is virtually absent on the seafloor. The chemical energy that sustains most deep-sea organisms is sequestered from sinking particular organic carbon, POC, derived from primary production in the euphotic zone hundreds of meters to kilometers above. However, POC flux decreases with depth in the water column because the material is remineralized and the distance seaward from productive coastal regions. A lot of scientists have researched on the energy on the deep seafloor before. Previous work indicates that although broad-scale patterns of deep-sea biodiversity are well established and presumably linked to POC, specific tests of this relationship remain limited. Here, we specifically use metabolic theory of ecology, an analytic framework to explore the relative contributions of thermal and chemical energy to examine the relative roles of carbon flux and temperature in influencing metabolic rate, growth rate, lifespan, body size, abundance, biomass, and biodiversity across broad taxonomic and geographic scales.
Results. The data are divided into 4 sets: metabolic rate, individual turnover and growth, biomass, abundance and body size, species diversity. Metabolic rate. Body size and temperature vary with metabolic rate for deep-dwelling taxa. The two groups exhibit no significant differences with respect to the exponent characterizing size dependence, the activation energy characterizing temperature dependence, or the metabolic normalization characterizing the size and temperature corrected rates. After accounting for size and temperature, metabolic rate does not vary significantly with depths. Thus, shallow and deep-dwelling benthic organisms are well characterized by a single metabolic rate model. Individual turnover and growth. Organisms that are smaller and living at warmer temperatures exhibit higher rates of individual turnover. Significant differences in turnover rate exist between data sources. Size and temperature both have significant independent effects on longevity. For deep-sea invertebrates, only mass is significant. All four datasets were characterized by a single mass exponent that is consistent with the MTE predict value of minus 0.25, and 3 of 4 datasets were characterized by a single activation energy. Overall, smaller species grow faster than their larger counterparts on a biomass-specific basis. Depth does not predict variation in growth rates. Biomass, abundance, and body size. After accounting for spatial autocorrelation, carbon flux, but not temperature, is a significant predictor of body size such that higher fluxes lead to greater average body sizes in a locality. For abundance and biomass, body size, temperature, and flux are all significant. Overall, greater abundances are observed at higher carbon fluxes and among smaller body sizes. The same finding holds true for biomass. Species diversity. The quadratic of carbon flux is significant for both Bivalvia and Castropoda. Thus, for mollusks, diversity peaks at intermediate chemical energy availability. Temperature does not predict variation in rheophile diversity in any case.
We find no evidence that the availability of chemical energy metabolic rates of deep-sea taxa consist with early work. Variation in carbon flux is the primary driver of patterns in biomass, abundance, and biodiversity. The exception is abundance and biomass, where temperature has a minor but a significant role. Although the size-temperature scaling relationship that we observe for rates of individual metabolism and turnover agree with each other, and the predictions of MTE, they differ from those relationships observed from growth rate. These findings are inconsistent because growth rate is ultimately constrained by metabolic rate and therefore should exhibit similar scaling. A potential explanation for this discrepancy is that the growth rate data compared here include both estimate calculated during some portion of ontogeny and estimate time average over their entire lifespan. The results suggest that individual metabolic rates persist as quickly as allowed by body size and temperature influenced biochemical kinetics, implying that chemical energy limits biota through its effect at higher levels of biological organization.